Hello, my name is Hyun-Sung Park. I'm the head of business development operation at Pharmapsin. Thanks for joining the session today, and I'm going to take it as an opportunity to introduce some of our assets that we are actively looking for partnership. Pharmapsin is a biotech company that is established in 2008. We are developing antibody therapeutics, and we are located in Daejeon, South Korea. That is about 1.5 hours away from Seoul by fast train. Pharmapsin is a public company listed on Kosdaq market with a current market capitalization of $235 million. Our lead asset is olinbesmab. It is an anti-VEGF receptor 2 neutralizing antibody. It has a best-in-class potential, and it is currently in 3 clinical studies. We have 2 potentially first-in-class preclinical assets that are being prepared for clinical studies in 2022. PMC309 is an anti-VISTA antibody, and PMC403 is a type 2 activator antibody. Again, we are looking for opportunity to license out olinbesmab as well as to license out or co-develop PMC309 and PMC403. So this is summary of developmental status. Olinbesmab is in phase 2 clinical study in Avastin refractory recurrent GBM, which occurs in Australia and in the US. We are finishing up 2 phase 1b combo trial with Keytruda. One study is an mTNBC patient, and the second study is an RGBM patient. And based on the interim data, we are going to continue on with a phase 2 clinical study in mTNBC. Both preclinical assets, PMC403 and PMC309, it's at the late stage of preclinical development. We are doing GLP tox study as well as finishing up CMC, and our plan is to initiate phase 1 clinical study in sometime next year. So I'm going to give you some of the summary of the key factors and the key results from clinical studies with olinbesmab. Olinbesmab has a clear safety profile compared to competing VEGF or VEGF receptor antagonists that are in the market, whether it's biologics or small molecules. And the results from a phase 2A study in RGBM patients showed 25% disease control rate and reduction of edema and tumor reduction in some of the patients. And one patient survived up to 525 days. And together with the data from phase 1b combo trial with the Keytruda in RGBM, we think that there's RGBM population, they do respond well to olinbesmab. Based on these results, olinbesmab received ODD status from US FDA as well as Korean regulatory agency. Currently, 12 out of 36 patients were recruited for Avastin refractory RGBM mono trial, and a phase 2 combo study with Keytruda in metastatic triple-negative breast cancer starts in September 2021. So I'm going to just show you the interim data from olinbesmab and Keytruda combo phase 1b study in mTNBC patients. There were 2 cohorts. The first cohort was treated with 12 mg/kg olinbesmab, second with 60 mg/kg olinbesmab. And the combo treatment was well tolerated. And there is one adverse event that was commonly associated with olinbesmab, which is hemangioma. Most of the time, it is grade 1 and grade 2, and it is reversible, meaning that once the treatment is stopped, hemangioma disappears. Efficacy-wise, if we just evaluate the second cohort treated with 60 mg/kg olinbesmab, 3 patients had PR. 1 patient actually has CR in the target lesion, but the non-target lesion remained. So this patient was regarded to have PR. So the ORR was about 50%, and DCR was close to 70%. The trial is going to end very soon, according to the protocol, and we still have 2 patients who are still treated in the trial. 1 patient received 26 cycles And second patient received 35 cycles as of the end of August. So I'm going to show you one patient data. So in here we evaluate the changes in tumor volume, and it is clear that the patient with SD or PR, they show a clear clinical response to this combo treatment. So based on this result, we are actually initiating phase 2 clinical study with this combo treatment in mTNBC patients. I'm going to move on to introducing two of our preclinical assets. PMC309 is an anti-VISTA antagonizing antibody, and it is an immuno-oncology product. VISTA is a broad-spectrum negative checkpoint regulator, which is highly expressed on myeloid lineage cell, including MDSCs, and it is expressed to a lesser degree on T cells, including Tregs. VISTA on MDSC is known to play a critical role for the MDS suppressor function. So antagonizing VISTA will block MDSC and thus relieve T cells from suppression, resulting in T cell activation. And there are evidence showing that the VISTA expression on immune cells from those patients who are treated with anti-CTLA-4 or anti-PD-1 or PD-L1 have higher VISTA expression. So it is speculated that the higher VISTA expression in these patients may contribute to the resistance and decrease of sensitivity to these immuno-oncology agents. So we are developing PMC309 to target those patients. They do not respond well to those agents targeting suppressor T cells, as well as develop combo treatment with other immuno-oncology agents, as well as cancer agents. So I'm going to show you two pieces of data, and this data came from ex vivo study. In this study, we isolated T cells and monocytes from human PBMC. We co-culture them and differentiate them into mature cells. And we activated T cells using anti-CD3 antibody, and we treated with PMC309 at the same time. So the data on the bottom shows that PMC309 treatment induced interferon gamma secretion, meaning T cell activation. And at the same time, the T cell in this the culture Treg population decreased. So this is a nice, very nice data illustrating and also supporting our working hypothesis. PMC309 does not cross-react with murine VISTA. So we engaged two animal models, and the data on the left side came from TNBC mouse model transplanted with human PBMC. So PMC309 treatment alone shows good tumor suppressing activity together with anti-PD-1 antibody. Now the tumor suppressing activities was stronger. We also used the human VISTA knocking model, which was injected with a CD26 colon cancer cell model. So the group which was treated with a control antibody shows continuous tumor growth over time. Now the group treated with either PMC309 or anti-PD-1, they start to show slowness of the tumor growth. The group treated with both PMC309 and anti-PD-1, now there's much stronger tumor suppressing activity. And when you evaluate with the tumor sample, there's increase of CD8 positive cell in tumor microenvironment in the group that was treated with PMC309, anti-PD-1, and as well as combo. And the MDSC population in TME were decreased by the treatment with the PMC309, anti-PD-1, or the combo. So that was quantitated and illustrated in this graph. So we are continuing the development and we are doing GLP tox and PK study in Charles River, and then we expect to finish the study by the end of this year, and CMC will be done by Q1 next year, and then our plan is to file IND sometime next year. So lastly, I'm going to talk to you some of the data, key data to test PMC403, a Tie2 activator monoclonal antibody. Activation of Tie2 is essential for vascular stability together with regulating VEGF axis. Tie2 activation leads to repair and normalization of abnormal blood vessels, such as blood vessel that's formed around tumor. So normalization of blood vessels can improve hypoxic condition in TME as well as delivery of immune cells. Due to this nice mode of action, we are exploring expansion of the disease indications. And currently we are testing PMC403 in ocular diseases, acute lung injury, as well as chronic kidney injury model. So I'm going to show you just a couple of data from mouse cancer model. The left side was tested, a PMC403 is tested in a mouse GBM model, and this is a great model to evaluate the morphology of blood vessels. So blood vessels around tumor microenvironment looks very thick and very aberrant. Treatment with the PMC403, now you can see the normalization of blood vessels and the shape became regular. On the right side, data came from CT26 colon cancer model. PMC403 treatment has mild tumor suppressing effect. Together with the anti-PD-1 antibody, the tumor suppressing effect is much stronger. So this tumor suppressing activity was associated with increased number of CD8 positive cells upon treating with the PMC403 as well as the combo with the anti-PD-1 antibody. So I'm going to show you a couple of data from eye disease model. So this data came from laser-induced mouse CNV model. CNV model is used as a model for human wet AMD. So when the eye was damaged by laser, now there are a lot of lichens, which was measured by fluorescence dye. Treatment with the PMC403 of Eylea, which is commercially available drug to treat wet AMD and DR, now can reduce the leakage area. And we also evaluate response to electric stimuli in ERG test, and both Eylea and PMC403 improve the response in this test. And we also test the PMC403 in the monkey laser-induced CNV model, and this data came from preliminary feasibility study. Again, the laser induced blood vessel damage and induced a lot of leakiness from the blood vessels. Treatment with Eylea and PMC403 now improve, repair the blood vessels and improve the leakage area. One interesting data came from ERG test. Even though there's a variability among monkey, we used 4 monkeys for this in this preliminary study. Overall, the response in the ERG test was much better with PMC403 treatment compared to Eylea. So this data was very encouraging, and we are conducting very comprehensive study in monkey, both CNV as well as diabetes retinopathy model. And we are expecting to gather the data by Q4 and getting the comprehensive report in Q1 next year. So we are preparing for the clinical studies in probably wet AMD sometime mid next year. So we are have done optimization formulation development for both preclinical as well as clinical study, and GLP tox and PK using eye injection is ongoing, and also the GLP tox and PK using systemic administration of PMC403 is ongoing. So thank you so much for your attention, and I hope that some of you may have found the data and asset appealing, and please feel free to contact me to further discuss about these assets. Thank you.